Hopefully we can continue the dialogue from this morning. And really a more appropriate title for this session is our molecular universe, because we're going to be talking about small molecules as well as macromolecular complexes and focusing on some of the chemistry, which we haven't really done before. And so I think some of the questions, how do we measure and define this universe? Networks versus mechanistic understanding of the molecules, and who are the participants and how do they communicate at the molecular level. And then signaling is the sort of dynamic framework that we have for all of biology, and not only signaling within cells, which is what I'll focus on, but signaling between cells, which some of the other talks will focus on. And then again, it's multi-scale thinking, going from the animal to the tissue to the cell, organelles, molecules, and atoms. So I think that's the scales that we're talking about. So I work on protein, I don't know if there's a pointer here or not. I work on protein kinases, and this protein phosphorylation is highly dynamic. So you have proteins being turned on or turned off constantly by this mechanism. And this is the kinome you saw before. It sort of began in around 1960 with the pioneering work of Krebs and Fisher, and then PKA was the second kinase to be discovered about a decade later. Then you had a whole flurry of tyrosine kinases being recognized. So these are the boundaries of this kinome family, and it's one of the largest gene families, and it's incredibly important for all of biology. And we've used sort of PKA as our model, our prototype for the whole family. And so, again, just to put this framework, and I would just say with Terry's synapse, he had, I think, 4 kinases and 3 phosphatases sitting there just in his preliminary model that he showed us. So, in this case, you have an extracellular signal, a hormone or a neurotransmitter that's binding to a GPCR, then generating cyclic AMP. And what we focused on is what cyclic AMP then does as a second messenger inside the cell. And its main target is PKA, which normally is dormant. It has regulatory and catalytic subunits. And it's only when you bind cyclic AMP that you unleash the catalytic potential of the catalytic subunits. And these are not, they're localized to specific sites in the cell, typically by these A kinase anchoring scaffold proteins. And so you create foci of PKA signaling in the cell. And so we've learned an enormous amount from these individual subunits. The catalytic subunit has told us about the features of the whole family, but it requires X-ray, but it also requires all these other techniques. You need to have ways to look at the dynamics. Now you can do simulations that are really at the level of the experimental data, so you can validate simulations. So we learn an enormous amount from the individual proteins, and these are networks that we really want to build. How do you integrate this community so that you can very easily communicate across boundaries? And so, like Terry, we've built collaborations with a lot of different people using this one enzyme as a model. And again, these are dynamic switches. They're not static proteins, and they're not metabolic enzymes. So now, though, if we look at how this protein exists in the cell, it exists as this complex. And these are 4 functionally non-redundant regulatory subunits. They are targeted to specific sites in the cell. And this is the physiological state of that protein, not the individual subunits. And that's really what we need to understand. So this was when we solved the first structure. And you had that tree, the beginnings of that tree at that time. But when you saw this structure, you really saw, okay, how does it fold in space? Those little white dots are conserved residues that are shared by the family. So you really went from a one-dimensional understanding to a 3-dimensional understanding. And you got a model for how every single one of those kinase cores folded up. So it's also phosphorylated. So this is an essential phosphorylation site. So you could see how one phosphate could make an extraordinary difference in generating this active enzyme. These are highly dynamic in how they are assembled. So you learned many things. This gave you an active kinase, and it showed you how one phosphate could make a great, great difference. So How did these proteins evolve, which we heard from Cannon? How do they work at the molecular level? How do they regulate biology? It's ubiquitous in every mammalian cell. How do they nucleate signaling nodes? How do they drive disease? So these are some really fundamental questions that the kinases are really important for. And so then if we step back and we say, okay, because they're so important for disease, we have many structures. And so these are solved by many different groups. And what can we learn from having a structural kinome as opposed to what we can learn from just delving deeply into one molecule? And so we look at the core. Sometimes mostly we have only the core. With Alexander Kornev, we discovered this concept of spines. And these are hydrophobic, we call them two hydrophobic stacks that are really fundamental in every active kinase, especially the regulatory spine. It's dynamically assembled, and it must be present for that kinase to be active. And so you can really use this fundamental understanding of this hydrophobic architecture of a kinase to apply those rules to chem kinase or the EGF receptor or whatever kinase you want. It's telling you really the fundamental principles of how those enzymes work. So if we look at it as a catalyst, then we look at it also as a scaffold, and that's the other function that the kinases have. They create a scaffold, and they use almost every part of their surface, so they're dynamically assembled scaffolds. And so that's how you build those networks, by adding phosphates and by creating networks of binding proteins. And so just to show you how we've learned about PKA, going from those individual subunits. In the case of the regulatory subunits, they have at their N-terminus a dimerization domain that's important in targeting, but then these two cyclic AMP binding domains. And this is how it's regulated. PKA is made as an active enzyme, and then it's immediately packaged as a holoenzyme, so its activity depends entirely on cyclic AMP. And so this is this cyclic AMP binding. Let's see if I can get my little pointer there. This is the cyclic AMP binding domain, so that's conserved throughout biology. It's a module that allows a cell, a bacteria or a mammalian cell, to actually take that cyclic AMP as a second messenger inside the cell and translate it into a biological response. And so if we now look at the next step, we had the individual subunit structures, but now we look at the complex. And seeing that complex was the first time you could understand how the catalytic subunit was actually inhibited and how it was activated by cyclic AMP. So it was an enormous advance in our understanding of that molecule. And so now I want to go on and just point out the importance of having not just that heterodimer, but the tetramer, what's the physiological state of the protein. And so what we've learned in the last 2 years really is now we're just going to pick up the pieces. Now you see here's the heterodimer. The blue is one cyclic AMP binding domain. The tan and white is the C subunit. So now you pick up a tetramer, you get a heterodimer, and for the first time you see symmetry in this molecule that you never saw before. It allows you a way to understand allostery. Then you pick up the second cyclic AMP binding domain. So this protein is activated by cyclic AMP binding first to the B domain and then to the A domain, and then it activates the catalytic activity. And so that's what Andrew was talking about, taking this model and applying it now to their understanding of the cardiac myocyte. How is cyclic AMP actually activated, and how does that relate to the model that they're generating? And there's a lot of wrong ideas that have just been used. And then you have the dimerization domain, which is going to be positioned at the top like this. So that's the little domain that's going to really involve this in targeting. And so again, just to give you a framework for how you put together a larger macromolecular complex, in circles is the dimerization domain, and the kinase holoenzyme. Sitting right next to it is calcineurin that we heard about from Terry. This is actually the same ACAP. It also regulates voltage-gated calcium channels. It sits there. Cyclic AMP activates the kinase. It puts a phosphate on the tail of the channel that facilitates opening of the channel, and calcium comes in. Calcium activates calcineurin. So you have this oscillation not only between a kinase and a phosphatase, between cyclic AMP and calcium, and this is the level where we really need to achieve that understanding. And this is accomplished in the case of the PKA targeting by this little 4-helix bundle module. The ACAP peptide is in red. It's an amphipathic helix that binds with a nanomolar affinity, and it takes PKA, so it's targeted to that specific site. And we have many of these complexes. So when we think about PKA signaling in the cell, we need to think about these macromolecular complexes where a particular isoform of PKA is localized in close proximity to a target protein, a channel, the ryanodine receptor, circa, and also close to a phosphatase. And very, very often that's calcineurin. So how do you then oscillate between calcium and cyclic AMP? How do you bring this dynamic parameter into our thinking about how we model these systems? So I think there are so many challenges, and one cannot do this. One has to do it as collaborative, working with people who are understanding the biology, having the computational tools to really bring different techniques together, and what we've been talking about here, modeling the whole cell. How can you extrapolate to whole cells? So these are just acknowledging the people that I've worked with. And I've worked very closely over the years with many different people in terms of trying to understand kinase function at both the static level and at the dynamic level. Okay.